It's really a pleasure to be here, and I thank Dr. Rampel for that wonderful introduction to all the work that many of us are very much invested in to try to, improve outcomes for all of our patients. And just as Dr. Rampel said, we're very excited about the future. I think it's brighter and brighter than it ever has been. And when I meet my young patients, I tell them my goal is to get you to 100 years. So I think, we're going to try to do whatever we can to keep your stem cells healthy and the rest of your body's healthy so that you can get there. And I don't think that that's an unrealistic goal. I think for many of us, given what we've learned about MPN and what we are learning, and with the new therapies on the horizon, hopefully that's achievable. So, there's a little bit of a play on words, new kids on the block. I'm referring to some of the new therapies and some of the new therapeutic targets, but also because I take care of children and young adults, there are kids on the block. Some of you in the field, at least my colleagues in science, know that I study a protein. It's called HMGA. It's a little small protein that's in all of our stem cells. And it's called HMG because it's small, and so when we run these proteins on gels, they migrate rapidly, and that's how it got its name. It also has these little hooks called AT hooks, which bind to chromatin, and you heard about chromosomal aberrations in patients with MPN. So these are some of those proteins that are latching onto the chromosomes and causing the stem cells to either self-renew and make blood, which is really, really important for all of us. And it also is facilitating some of the mutations that our MPN patients have to make too much blood. So they have a very interesting role. They're present normally, but they can also be overexpressed when our patients have MPN mutations, such as JAK, which of course is the most common mutation that's present in our patients, but other mutations. And just as you heard from the beautiful talk by Dr. Rampel, we're just learning about those lesions. And something that's also very exciting in the field is that sequencing technology is becoming less expensive. Before, we used to just sequence the coding genes, but now we're learning about used to be called junk DNA because no one knew what it did, but it turns out so much of our DNA that doesn't make proteins is actually regulating how the protein coding genes are expressed. And so that's a super exciting area. And when we talk about familial predisposition, for instance, we're finding that some families have alterations in this so-called junk DNA that's predisposing them to MPN. So that's why, Dr. Rampel said we're confused because we don't really know the whole picture of all of our chromatin and all of our DNA. It's huge, but we are learning. There's some very large studies coming from the Broad Institute, which is here in Boston, where we are looking for lesions within this non-coding region of our genome that predispose to MPN. And in fact, one of the more recent studies implicated the HMGA loci as being associated with increased risk for MPN. So I think, in answer to the question, is this familial and what do we know? This is very much an exciting and emerging field, and every year we get together, we will have more to tell you about. But we were excited when the Broad study showed that alterations in the loci for the gene that my lab has been studying for decades is associated with an increased risk. And I guess I wanted to say one other thing. This paper was provisionally accepted for publication where we looked at our Hopkins cohort, and we've been following patients way before I even was at Hopkins. I've been there over 30 years, but we have a cohort of over 600 patients. And some of them were diagnosed in their 20s and reached their 50s and 60s. And so it's an interesting cohort because for many of those patients, we know what their mutation is, and we also know what happened. And I guess there was a question I'll just follow up on, since it's at the top of my mind, was, well, what about these pediatric patients? Are they progressing more slowly? And I think that's a great question. And as a pediatric hematologist oncologist, that's the question that's on the top of my mind. And one thing, and again, this will come out probably in a few months in the British Journal of Hematology, but one of the things we looked at was the length of time for progression, and it was indeed slower in our pediatric patients. So I think that's comforting, but as doctors in the field and scientists, we don't want to see progression at all. And so that's really the focus of my lab, and I think the focus of everyone in this room. And so these proteins, as I mentioned, seem to be playing a really important role, and I'll share some of our human data and some of my little tiny mouse patients and a little bit of a few words to share on my young patients. But these proteins kind of function by opening chromatin to enhance gene expression. And you've already heard about some of the abnormal genes that get expressed when someone has JAK mutations. And as I mentioned, I've been studying these genes and proteins for decades, and they're really fascinating. They're even present in plants, and they mediate a plant's response to sun and wind, and they're in our blood stem cells. And as I alluded to, they get cranked up when people acquire JAK mutations, and even other mutations associated with MPN, which you heard about from Dr. Rampel's talk. There are other genes that seem to alter the course and the progression of MPN. And we actually first showed that these genes, when you overexpress them to very high levels, can make blood cells grow like leukemia cells. And so we're very much focused on understanding exactly what's happening at the cellular level, because we want to intervene and prevent that. And this is just one example of some of our mouse models that get leukemia. And this, like many important genes and pathways, even JAK-STAT signaling, it's not only important in our blood stem cells, but it's important in many of the cells of our body. And the little green cells there, they look a little bit like a Christmas tree. They're actually in the intestines, and there's stem cells in the intestine. So some of the same pathways that are active in our blood stem cells in MPN are also active in our gut stem cells. And that's, and they're active in other stem cells. So that's one of the things that as physicians treating patients with stem cell disorders, we have to be very aware of what these pathways are and where these pathways are active, because you can imagine if you shut off a pathway that's important in blood stem cells that is contributing to MPN and making too many blood cells, you have to think about, well, wait a minute, if that's expressed also in the gut stem cells, we don't want our patients having GI toxicity from their medicine. So we really have to take a very comprehensive view and not just look at the stem cells in our blood, but we have to be aware of all of the tissues of the body that may have these pathways. And that's why some of our treatments affect other organs of our body. And so I think as you heard earlier, the most common mutation is a JAK mutation. And I kind of drew out the pathway. The little pink balls on the top are, cytokines, and these are inflammatory proteins. And they also tend to increase with aging. And so that's one of the ideas that many of us are studying. With aging and with increased inflammation, this can help these mutations expand and progress. And just like Dr. Rampel mentioned, something that is super exciting in the field is that we are tracing these mutations back to in utero, which I just think is a, knock your socks off discovery, because what that means is, these mutations are present from the very beginning, or very close to the very beginning, and something is causing them to expand and to cause a disease phenotype. And I remember Anne about 4 or 5 years ago mentioned, this is going to be more common in pediatric patients. I just know it. And I thought, well, I'm just not sure how that's going to happen. And she's right. And it may not be that there are more patients, but now that we have improved technology to identify these genes, we're seeing more patients. And not only improve technology to diagnose it, but also to figure out in our 40 or 50 or 60-year-old patient, when did they acquire those mutations? And I think, it's actually an optimistic finding, because if it's really occurring that early, that means there was something going on that prevented it from expanding and causing a blood disease. And so, as a pediatrician, that's the focus of my laboratory and my clinical care, is how we can prevent these things from causing a disease. And it's always easier to prevent something than it is to treat. And so I think these studies looking at mechanisms of stem cells is really exciting, and I think that's where we're going to find ways to intervene and prevent this hyperactive JAK-STAT signaling. And so here, this is just a picture showing that when patients have these mutations, and whether they happen in utero or sometime later, over time, the number of mutant stem cells tends to expand. It's like there's a fitness advantage. And, you know, you think of fitness as a good thing, but in a stem cell hierarchy, it may not be the best thing. Like, you don't want your mutant stem cells to be fitter, you know, to outrun and outcompete and expand above the wild type. And so that's another area that many of us are studying with great detail in the laboratory. How can we exhaust those stem cells and cause the normal stem cells to expand? And, you know, just like, oops, my pretty little pink circle didn't end up where it was supposed to. It's supposed to be at the bottom. And, you know, just as you heard about, you know, we would like these diseases not to progress. In fact, honestly, I wouldn't mind if we knew they occurred in utero, but I'd prefer them never to manifest as a disease. And I think that's where all the excitement is in the field, to study what exactly is changing and why these mutations are expanding and causing a disease. And so now here, this is actually human patient data. So when we were interested in learning whether this HMGA gene could be important in MPN, and we thought it would be, because I showed you in my laboratory, we'd already known it's a really important stem cell gene for stem cells to make blood, and our blood stem cells are super duper stem cells. We have to make billions of blood cells every day. So you can't have a wimpy stem cell doing that, or you'd never, you know, make blood for 10 years, let alone 100 years. And so these stem cells are incredible stem cells. And we knew that our gene and protein were enriched in stem cells, and that they endowed the cells with the ability to what we call self-renew, which means make an identical daughter cell, and also to make all the blood that we need to exist. And so to determine whether this really could be contributing to MPN and MPN progression, we just measured levels in blood cells. And this is some samples of CD34 cells, which are enriched for stem cells in our patients. And you can see the little dots are going up higher and higher. And so we found that in normal stem cells, it's enriched compared to, like, you know, differentiated tissues. But when you develop PV, it goes up higher. And then as you progress, it goes up even higher. And we looked in a number of different cohorts. This is, I think the middle one is a cohort that Dr. Rampal was involved with. One nice thing about the MPN field is all of our colleagues are incredibly collegial, and we collaborate all over the world, and it's really been wonderful and important to advance the field. But these little dots on the end there, they're a friend of mine in the UK at Oxford, and in the UK, they have been pioneers in sequencing technology, and they sequence a large number of MPN patients. And so, and they're also generous and share data, and we can see that in these, this is single cell, so now technology has advanced such that we can actually do sequencing in a single cell, which is really quite a feat that none of us anticipated years ago. But when you look at the single cells, we're seeing that this gene is in fact increased. And even, I think, more intriguingly, we can look at MPN patients, and that's what this last chart shows. We're looking at MPN patients, and as you may know, some of the stem cells are mutated and some are not. And what we did in this study, together with our colleagues in the UK, was we looked at the mutant stem cells versus the not mutated stem cells. And we found that HMGA is actually up preferentially, exclusively in the mutant cells. So that tells us that, you know, there's something really important going on in this mutant context. And we're very interested in dissecting what that is so we can prevent those, the bad guys from expanding and keep the good guys active. And so, you know, when we look at function, now we actually have all kinds of exciting technologies that allow us in the laboratory to look at function of genes. Like you've all heard about CRISPR, super exciting genetic scissors where we can cut out genes, and we've done that in blood cells and in mice. We can silence genes using different approaches. And so when we were first trying to figure out what this gene might do in MPN that's progressed all the way to leukemia, we used those techniques. We used genetic scissors or CRISPR. We've used something else called short hairpins. And when we use those approaches, we find that it makes those mutant cells stop growing. So that is something we're really intrigued by, because if we could make mutant stem cells in our patients stop expanding, I think we would be able to make a big impact. And so this just shows some of the experiments we do. We cause the cells to grow less fast, and they're shown in the red and the blue lines. We cause them to not be able to grow we have an assay that's called clonogenicity. It's sort of a leukemia growth assay, and we show that we can prevent that. We put them into mice, and these bone marrows look similar to some of the bone marrows you saw from Dr. Rampel's talk. We can put leukemia, some of my colors aren't coming out, but what I wanted to show you is that when we silence this gene in leukemia cells, the leukemia cannot engraft in mice. It can't grow. And also the mice live much longer. And even the mice that live longer and ultimately get leukemia, what we found is that in their cells, they found a way to escape gene silencing. So they absolutely required HMGA to expand their mutant stem cells and to develop leukemia. And we're looking at other drugs in the field. In this case, we looked at ruxolitinib. It's actually turns out to be an easy drug to study in the lab because they actually make mouse chow that has it mixed in, so you can give your mice this drug and see how it interplays with the genes you're studying. And we found that together, those two treatments, lowering the expression of this gene and Rux, worked together to help prevent the leukemia from growing and to expand survival even longer. So that to us wasn't so surprising. We thought, well, we know this gene causes leukemia. We thought it probably would be important in MPN leukemia because it's important in virtually every aggressive leukemia we've looked at. But then, that's sort of after the cow's out of the barn, I guess is the saying, or the whatever. That's when it's too late. We want to get in earlier. And so, cat's out of the bag, I guess that's one of the other sayings. So we wanted to look earlier, and so it turns out there actually are really beautiful mouse models for early MPN. So these mice get elevated blood counts, just like our patients, and they can even progress to fibrosis, and you saw the beautiful bone marrow examples of that in the talk before. And so these are just blood counts in our mice. The blue ones are normal. The orange ones are the MPN mice, and you can see, just like our patients, their red cell counts are higher, their hemoglobins are higher, and their platelets are higher. And we can cross them to mice that have just a half a dose of our gene, and those mice are actually super mice. They live extra long because they're protected from tumors. And we were thrilled, and this was surprising because, again, we thought of this sort of as a gene that really is involved with latent disease. But we found that if these mice are engineered such that they have just half a dose, they don't get myelofibrosis. Like, these are the spleens, and they sort of look like human spleens, but a lot smaller. And you can see the ones that have a half a dose on the bottom are completely normal in size. They're the exact size as what we call wild type, the normal ones. And the JAK spleens are bigger. And then when we look at the marrow of our mice, you can see they really resemble what you saw in the patient samples. The JAK mice get too many megakaryocytes, and the last two, I'm not sure how you can see this. I'll try to point it out with the arrow, but this is fibrosis. And that's the thing we want to prevent in our patients, because when the marrow starts to get fibrotic, it's almost like a scar is there, and the blood stem cells are going to have a lot of trouble making more blood because the scar is getting in the way of how they function. And so with our mice that just have a half a dose of HMGA, their marrow is staying very, very normal. And here is just a kind of a far away view of the marrow. This is a normal mouse. This is a mouse with myelofibrosis, and look at all that scar tissue in there. And then when we take away one of its HMGA genes, it completely prevents that from happening. And then we do transplant experiments in mice to sort of see, this is a nice experiment because you can kind of compete the mutant stem cells with the wild type stem cells and see what happens. And you can also do experiments where you're just looking at the stem cells. And we saw the same exact data. If you take away one dose of HMGA in the blood stem cells, they can't develop fibrosis. And so we are really excited about that. And we do some fancy procedures called flow cytometry, where we can look at what kinds of stem cells are changing. There are some stem cells we call long-term, short-term, and progenitors. And that helps us to understand where this pathway is working. And we also more recently have started to do single-cell RNA-seq. I know Dr. Rampal's lab is using that, and that's a really super exciting technology because, like I mentioned before, it allows you to look at the genes that are expressed in the mutant cells. And so in this case, we're looking at the genes expressed in each cell in all these different stem cells and some of their progenitors, and we're looking at what happens in the MPN mice and what happens in the wild type mice. And we're just starting to dig into this data. As you can imagine, we have 30,000 genes, so there's a lot of information to dig into, but we're finding some really exciting pathways. Like we mentioned, with aging, there can be more inflammation, and we're finding that to be an important pathway. We're finding some genes that may drive fibrosis, and that's really exciting because that can lead us to how we can prevent those abnormalities from progressing. And we also do sort of what we call bulk sequencing, where we look at all the leukemia cells to see what the pathways are. And this is just an example of that. And I won't bore you with the details, but one of the super exciting things we learned from this study was that immune pathways are being turned down with progression in this model of leukemia. And you guys, I'm sure, have heard about the excitement in the field of oncology with immune therapy, right? It's revolutionized some tumors, but not most. And so we would like to harness that approach. And one of the things we're learning is that when these cells that are fibrotic start to become leukemic, they're shutting down their immune markers. And so that kind of allows them to expand and grow and be invisible to the immune system. And so we found, and we're starting to sort this out, and a terrific grad student in my lab will be giving a talk at our hematology meeting in December, but we found that the HMGA protein is helping to dampen these immune activation response. So it's sort of making these mutant stem cells kind of stealth. They're not seen by the immune system. And what we'd really like to do is turn on those immune markers and get our own body's immune system to attack them. And those are some of the pathways we're currently teasing out, and I think that's looking really promising. I'm just going to skip through this because it's a little bit basic and probably not so exciting. Oh no, and I guess I lost a little bit of the background here, but this was a really wonderful collaboration that we did with Dr. Rampal, and he has this very unique collection of patients who had fibrosis and then developed leukemia. So you can imagine this is a wealth of data. And, like I mentioned, our community is very collegial and generous with sharing data. And so we looked at the pathways we were interested in these patient samples where very deep sequencing was performed. And we found, because the stuff I've been showing you, well, sure, it's nice to cure MPN in a mouse, but really that's not what we're after, right? We really want to help our patients. And so we always go back to the patients after we dissect pathways in our experimental models to see, well, do they happen in our patients? And indeed, we found that the gene pathways we observed in our mouse and cell models were being activated in our patient samples. And so that gives us even more impetus to drill down and try to prevent those pathways from being activated. And here's some more samples. Again, these are from our colleagues in the UK at Oxford, where they're doing single cell analysis of many MPN patients and also healthy age-matched controls. And we're finding that our gene is activated in some of the downstream pathways. And so we're excited about this data, but we really feel like this is just the start, because obviously we don't want to cure MPN in mice, we want to cure it in patients. And one of the things, I'm just going to breeze through some of this more molecular stuff, but and we were really excited. Our MPN work was featured on the cover of Blood, and you guys will recognize this as the marrow from the mouse that had fibrosis. I actually wanted to show the normal one because I thought that's what's exciting, show the fibrosis, but they said no, we can just show one picture. So I thought they kind of took away the best part of the picture we asked them to use, but that's what they could do. So, and then I just wanted to say, one of the things we were surprised at, and this paper will come out soon in a journal called JCI, but some of these same pathways that are driving fibrosis in the marrow are also driving fibrosis in other tumors like pancreatic cancer. So what we learn in MPN probably isn't just going to help our MPN patients, but some of these pathways are going to be relevant to patients with diverse tumors. And so now to the new kids on the block part. So we are looking, as you can imagine, for ways to block our HMGA protein from causing leukemia and fibrosis. And we were introduced to an investigator at the Icahn School of Medicine who's developing a new technology called PROTAC. And it's exciting technology. There are some in the clinics, none so far for transcription factors, but the data is looking super exciting. And the idea behind these molecules is that they attract the target, the bad guy, and then they degrade it. So it's kind of like, a little decoy to trap the protein you're trying to target, and then it targets it for being destroyed in our cells using our body's own protein destruction mechanisms that are normally present to get rid of proteins that our cells no longer need. So it's a nice approach because it harnesses what our cells already use, but it targets the bad guys. And so we have some promising data. It's still early days, but we're excited about that, and that's definitely a new kid. And there's also another interesting PROTAC that was just published, and this came from Dr. Hoffman's lab and his group there, the same group that we're collaborating with, and they're targeting another protein called MDM, which is important in a P53 pathway. And Dr. Rampel's lab has studied P53 and how that plays a role in leukemia. So I think that's another exciting target, and hopefully there will be more to talk about in years to come. And as Ann mentioned, I focus really on younger patients, and we have been defining thromboses. One thing that's still quite perplexing to the field is that many of our younger patients are getting some unusual clots that actually affect their livers. And it's more prevalent in females, and it's also more prevalent in younger patients. And that's still a bit of a conundrum because you kind of think of a younger individual as having less, having healthier tissues overall. They haven't had inflammation, there hasn't been any vascular injury or damage. And so it is quite surprising that these patients are at risk for this particular type of clot. And we're doing studies to try to understand that. We're also doing studies to try to prevent that. And one of the therapies that we're very excited about, and we have some of our pharmaceutical colleagues here who have been helping with these, but there's a drug treatment called interferon, and again, we're not completely sure how it functions, but some of the ideas that have been proposed are that it's targeting the bad stem cells. It's causing them actually to self-renew, which you think, well, that might be a good thing, but then like other stem cells, if you self-renew, there's a certain number of self-renewals that all of our cells can do in general, and then they exhaust. And so what we'd really like to do is treat our patients, get the bad guys to do whatever they need to do to exhaust. And we're excited because we've been starting to plan and orchestrate a study for our young patients that we're hoping will, really change treatment for younger patients and older patients. And even though I'm a pediatrician, older patients are very near and dear to my heart, and this is actually my mom, who also has MPN. She's like 86, she walks 2 or 3 miles a day, she's got JAK mutant disease, and she's doing great. And it's interesting because I think like in her 40s, I remember she got diagnosed with a migraine, which was, she never had migraines before, and she went to the hospital, and I remember looking at her blood counts, and the hemoglobin was a tiny bit high, and I was a little concerned. I thought, could this be an MPN? And I showed it to my MPN friends who treat older patients, and they all said, oh, they look okay to me. Well, lo and behold, 20, 30, 40 years later, she really does indeed have this. And I think it goes back to what we were talking about. Patients are developing these much earlier than we ever thought. I mean, we don't know, but it could have been there in utero. It could have been there when she was 20, and then something caused it to expand. And so, we would like all of our patients to get to 86, 96, and to be healthy like this. And so we're very excited about, dissecting those molecular pathways and then discovering ways to prevent them from happening. And that's my dog. And these are our funding that we're thrilled to support this work. So hopefully I gave you a little snapshot of what we're working hard in the lab to do to help all of our patients.